Hi, hello everybody. My name is Christian. I'm a research scientist here at the Institute for Systems Biology. I'm super happy to see all of you here, and I will be the one that will walk you through the next 2 days of courses that we have designed for you. And we will start today by looking at Amplicon sequence data analysis with Qiime 2. And it's basically like a standard pipeline that's used those days to analyze mostly data from Amplicon sequencing data analysis, in some cases also for metagenomic data analysis. And since we still have a little bit of time, let's kind of introduce the motivation for the course a little bit. So it's mostly aimed at basically getting you up to speed from getting raw sequencing data from any sequencing provider or your own core unit, and then going all the way to generating some kind of scientific insight. And like I said, today, we'll be looking at how to basically analyze like community data from Amplicon sequencing data. And tomorrow we'll actually be looking a little bit more at bacterial isolate sequencing and how we can actually generate some functional insights from that. I should also mention that the entire course is actually designed to be viewed asynchronously, meaning that you don't really you're not limited to participating here only live. You can actually take this course whenever you want to, whenever you find the time. That's why we actually have the Slack for that, where you can actually ask questions at any time of the day or night. And receive answers, and that will remain open for at least like 1 or 2 weeks after the course. So if, for instance, that doesn't work for your own time zone right now, or you would have to get up in the middle of the night to catch the live sessions, don't worry at all. You can simply see those recordings, which will be made available pretty quickly after the course ended. And you will have the chance to kind of go through the materials, run all the code at your own time, and still ask questions in the chat, like the same way as we do right now. So there's actually no limitation of not taking the course right now. So you can do that at any time that fits. And just to start off a little bit, the slides as well are available, and they're basically just like a website. So you can basically open that on any device that can open websites, which those days are quite a lot of them. So you can open it on your computer, your phone, your TV, your smart fridge, if you have one. Anything that has like a widescreen screen can basically be used to view the slides. So feel free to open those also on your phone, like to the side, if you want to go back and forth. Since we'll be using like a few different websites today to kind of run the course. So you will switch a little bit, but for most of the time you will probably be using the notebook and following the Zoom call. But you can open the slides anyway if you want to go back to a particular slide and check out some materials again. So now kind of getting to the official starting time, let's talk a little bit about the organization of the course. Like I said, it's like kind of a 3-tiered approach that we're having here. So basically, I will be running through the presentation here, which is mostly serves to give some logic, explanations, links, like anything that's like background information that might be interesting to you. The most of the actual action will be happening in the interactive notebooks that we'll be introducing in a few minutes. That's where we will be actually running through the analysis. You will get the data there. It will explain all the kind of technical aspects of the analysis. You will get links to materials, and you also generate visualizations directly there. And you don't need to install anything. That just also works basically in the cloud. We're using Google Collaboratory for that, which basically gives us a mini compute server in the background that you will connect to. So everybody will have like access to exactly the same resources. And yeah, so it will all be fairly standardized, which is nice because we can definitely help you out better that way. And then maybe also important is the chat that we have in the Slack format, where you actually can receive support and also, ask questions and answers. And maybe one thing that I should mention is like for the course, please change to the course channel. That's kind of what we primarily monitor for the course. The general channel is kind of if you want to say hi to everybody, or you want to just talk about the microbiome because, let's be honest, the microbiome is awesome. So you have the course channel there where you can actually ask really targeted questions, about anything that's happening in the course. And as you see, we will also have a channel for the symposium where you can, have some background discussions, but more about that on day 3. But yeah, that's kind of the distribution today. So the way we'll mostly be working today is that you will have the notebook open, because that's where we'll do all of the, the hardcore data analysis. And then you will have the Zoom presentation maybe to the side or in the background, or you can also switch, where I'll be explaining basically what's happening. But yeah, everything that we will run will actually happen in the notebook. So let's maybe look a little bit how that actually looks. And let's get started a little bit with understanding how that notebook works and, what is it all about. So if you click on that link in the slides, and I'm pretty sure one of the TAs will probably also post it in the Zoom or in the Slack as a link. So that's kind of an interactive notebook. And like, one way to understand it is, well, depending where you're coming from, if you're maybe coming from the wet lab, you could actually think about it. That's like kind of like a lab journal or protocol, but it's actually like a supercharged protocol where instead of just, annotating what you're doing, you can actually also run it in the protocol. So if you're basically saying, do that experiment, it will actually run it within the notebook itself, which seems like crazy magic, but well, that's what we're doing here. And if you're coming from another background, you can basically imagine it like an interactive document. So it's like a supercharged document where we obviously have text, but we can also like integrate code and actually run it and have it executed in one of those servers that are connected to the notebook. So let's maybe like look a little bit on how that environment looks like and how it works. So you open that notebook, and actually the first thing that we probably want to do is to create our own personal copy of it. Like, you can actually run the one here, but it's not a little bit less convenient because it will actually not save everything for you for later use. So if you want to come back tomorrow and actually see what you did today, you probably want your own copy. So to do that, after you open the notebook, you can go here on the file thing, and you can basically save it in your own Google Drive by saying save a copy and drive, which is like the first option in the save options. So if you do so, it will ask you for a sign in. Maybe let's do that from my side here. And just, you will still hear me. I'm just gonna remove the video for a second. And it might ask you for the same thing. Oops. Oh, let's take a short break here. There is some authorization I need to do. Sorry about that. Okay. Okay, we're back. I'm sorry, that's probably much easier for you than was for me. So basically now I logged in, you see my screen again. So after you have authorized, you can basically save a copy in your own drive. It will create a copy. It might ask you to rename it because it's like not a super cool name. So I might actually say Christian's notebook here. Okay, and now you have your own copy, and everything that you execute here will actually be saved. So let's talk a little bit what that environment looks like. Like I said, it's an interactive document. So you have something which is called cells. Cells kind of like separates a document into individual parts, or really tiny parts. So like the first one, for instance, you see if I hover over it, it's basically just text and it has some links and background information. But you also have those other cells here that have that little play button to the side. And those are code cells. So everything that is in here can actually be executed on that computing server that's connected to that notebook in the background. So if you like push play, it will actually execute things. Then on the side here, you have like a sidebar, which like first has a table of comments. It has some search where you can find stuff in the document, but it also has a little folder symbol where you will actually see files as they appear. If you click on it, maybe automatically it already connected you to the backend. In case it didn't do it and you still have that connect button here, you just can click on it and will actually connect to the backend, which will be pretty quickly. So now, for instance, in my case, and it will look the same for all of you, you kind of see that we're connected to like a compute server in the background, which is not like, you know, a super powerful machine, but it's enough for what we will do. So we kind of see that as like 13 gigs of RAM, and we have like 100 gigabytes of disk space here. So that will be fine for us. So now where we're connected, we can actually run those cells and execute code in here. So let's maybe go to the first cell here that actually pulls in all the materials for the course, and let's run this. So that will basically download all the materials that we need. And if you would look again into that folder symbol here, you actually see that we get that materials folder here that has like things like the raw data, also the notebook, and a lot of other stuff. And we will actually be working only from within that materials folder. So the first thing we do is actually switch in there. So that's like the second cell that we will be running. And it told us that we changed into the materials folder. There's nothing really that's happening here. The only thing is that everything we execute from now on will assume that we're operating within that materials folder. So it will see all those files. And then the last thing we'll be running for that very moment is the third cell here, which kind of runs the Qiime 2 setup. And what that will basically do is install Qiime 2 into your environment and set up everything so we can actually run Qiime 2. That will take a little bit, so about 10 minutes. So that will be like a good point to kind of go back and actually talk a little bit about what we want to do today. But yeah, you can just like observe that. That's like, that will just run for like about 10 minutes and install everything. Maybe like as a troubleshooting tip, since those are like kind of free compute servers connected in the background, if you kind of leave that notebook alone for too long, it might disconnect you. As long as you get a button that says, you know, reconnect, you will actually not lose any files, and everything that's installed will still be installed. It will just reconnect you to the background. To the compute server. So if you ever get that, you can just say reconnect and continue wherever you left off. In case you accidentally, for instance, close the tab and you close the notebook, it might actually, you know, start up the notebook anew. And in this particular case, you will have to rerun everything. But if you have any problems, please feel free to ask in the Slack chat and our TAs will help you out. Okay, so that will run the setup here, and it will take a little bit because Qiime 2 has quite a bit of software included. So let's maybe continue with our talk and the slides a little bit during that. So we did our setup, and maybe that like all seems a bit too much, like it's a lot of overload of information here and we're all running those steps. Maybe at some point you feel like, you know, it's hard to keep up. Don't worry at all, like don't panic. Like you can actually go through it at your own speed as you want. And also all the output that we generate can also be found in the treasure chest folder. So there's a link here basically in the slides. So if you click that, you actually see all the files that we will generate. So for any reason, for instance, like we can't really access Google Colab, or there's like some limitation that keeps you from running the code live, you can also go through that treasure chest, because at some point we're going to look at visualizations that we generate, and you can also download those from there and just look at them. So that's like an alternative, like a backup if something doesn't work as we expected, which, you know, we already had a few instances mornings, but well, we fixed it all. So let's maybe start and talk a little bit about Qiime. Like, why are we actually using Qiime to analyze 16S Amplicon data? Because, you know, there's a lot of other ways to do it. So first of all, it's pronounced like wind chime. So that's where like how you pronounce that name. It was created around 2010 during the Human Microbiome project that ran from 2007 to 2016. The main leadership of Qiime design is still with Greg Caporaso even today, and it was done within the group of Rob Knight. And I mean, you might have heard of Rob Knight as one of the major persons operating in the microbiome field, basically. And basically, it came out of the motivation that they observed that, you know, analyzing microbiome data is actually a bit more complicated. Like, there's a lot of steps to it. The experimental protocols is one part of it, but then actually analyzing the data is also fairly complicated. And basically, the major complication is that where we usually analyze like a single organism, here we're actually looking at like an entire community of organisms in an environment. So there's like DNA from all those different organisms mixing up, and we have to kind of like pull it apart, see who's actually there, and it's actually quite challenging. So like they notice that you have those long analysis pipelines, and you might have seen that before. Like at some point in that pipeline, it becomes really hard to keep track. You have that like that intermediate output file you generated a year ago. You have no idea how you actually got there. You know, you might have run like a combination of 5 different softwares to get to that file. You have completely forgotten what the parameters are and so on. So they thought about that, and they designed that like as kind of a one-stop solution to kind of do like most of the basic analysis for microbiome data. And that's kind of the mission of Qiime. And then pretty recently, like Qiime 2 was released, and it was like actually a major change to how Qiime 1 operated. It was actually had a bit more emphasis on data processing, but also analysis transparency and tracking how you actually got to data. And Qiime itself actually means quantitative insights into microbial ecology. So that's what you can do with it. So what is it actually? Like, you know, it seems quite cool to say it's like a pipeline or it assembles pipelines, but basically it's a set of comments to transform microbiome data into intermediate outputs and visualizations. That's what it actually does. So like we have that little GIF here that shows you like an example how that would work, where you kind of can generate, like you have some input data and then you generate output data. It's commonly used via the comment line. And the comment line is basically a way to talk to a computer. That's how you could imagine it. So basically you can write like in text what you want the computer to do and it will do it. And that's how it's mostly used. There are interfaces, so like different interfaces, so but they're a bit in progress right now. And Qiime 2 was introduced in 2016 and improves upon Qiime 1. And there was basically 2 major challenges they encountered like during Qiime 1. One is that in those long-running pipelines, it's sometimes a bit hard to see like which input data you can actually use for a particular analysis. Like sometimes it's kind of obvious, like if you have something that needs DNA sequences, you can't really put in like counts of organisms or something in that particular analysis. But sometimes you might have counts, sometimes you have counts that are kind of normalized into percentages, for instance. So is that still is that still applicable for that particular analysis and things like that? So one thing they did is like they had in Qiime 2 is they have those type systems that can actually check if the input to a particular analysis is actually valid for that analysis. So they can basically validate if whatever you put into an analysis is actually, you know, the correct data to use here. They also have that integrated tracking of data provenance, which we'll have a look a little bit later on, which is really cool. It basically allows you to look at any output generated by Qiime 2 even years later, and it will tell you exactly how you got from the raw data to that particular output, including all parameters that were used, individual steps and programs. And that's like super helpful, especially in a scientific setup. You might have all run into that situation. You know, somebody in your lab did something a few years ago. Now you need to go back to that analysis. You get some output file. You know, you have no idea how you actually got there. You might want to use the same analysis for new data, but you actually don't know the individual steps anymore. And like Qiime 2 can really help you with that. It also has an extendable plugin system. So there's a lot of people that develop plugins for Qiime 2 to do additional analysis. We have done so, for instance, ourselves. And like last year's course, we introduced a few of those. And there's many others. So it's kind of actually quite nice because, you know, if there's like new papers coming out with analysis methods, a lot of them will, for instance, provide them for Qiime 2. So you can basically plug them in with your own data analysis pretty easily. Okay, where can you find help? Like Qiime 2 has an extremely great community that is not only super helpful in the user forum, but there's also like a wide range of tutorials, general documentation, and there's also like a set of forum where you can ask questions. They're super nice. They will reply super quickly to you. And there's so many people that have like so much background and experience that you usually, you know, get a really good solution for whatever you want to do with Qiime 2. So it's like really, I think it's really friendly if you're like entering the field as well, because you have, you know, that really welcoming community that will help you out. So let's talk a little bit about how Qiime 2 works, since we'll be using it today. So the basic idea behind Qiime 2 is that you have something which is called artifacts. An artifact sounds like, you know, we're not going into archaeology here, but basically it just means it's intermediate steps in your analysis. So you could, for instance, imagine that you start from raw sequences. That for Qiime 2 would be an artifact. And then you transform those sequences maybe into, you know, some clustering of the sequences or maybe counts of organisms, and that would be another artifact for Qiime 2. But at some point, you probably want to generate like a figure or like a table with abundances, like something that you actually can look at and understand what's going on, and which will actually not be transformed anymore. That will be your final output. And that's actually called visualizations. And to go between those things, between different artifacts and between different visualizations, Qiime 2 basically uses actions, which are basically analysis steps. So like, here's, for instance, an example on the right side, like for pretty complex analysis where you start with raw sequences, and here's the green things, the green like rectangles, rounded rectangles are artifacts, the yellow rectangles are actions, and then the blue rectangles are visualizations. So we'll start with an artifact, which are your raw sequences. Then you might demultiplex them. Demultiplex basically means if you had several samples in one sequencing run, you basically split them up into sequences for individual samples. So you get your demultiplex sequences as an artifact. Now you maybe do a denoise cluster analysis, which gives you feature tables, representative sequences, and we will have a look what that actually means. And there's like a lot of different steps. And then at some point, you might actually say, hey, now I want to run a differential abundance analysis to see if one microbe is more abundant in a particular condition than in another. And that would be like a visualization. So a visualization in Qiime 2 is not necessarily a figure, something you can look at and that has colors. It can also be a table. It can be a statistical test, which has a p-value attached to it, something like that. But that's the main idea. And it's also summarized here on the left. So you have the artifact, which is kind of like transformed. So the basic thing here, I think, or the take-home message is like to remember in Qiime 2, artifacts represent intermediate steps, but visualizations are endpoints meant for human consumption. That's basically how you can differentiate. So whenever you want to generate something that you look at and you interpret, that's probably a visualization. An artifact is usually something you don't actually look at, yet you need it, you need to generate it, you need to run that analysis step, but it's not something you actually look at. And Qiime 2, like I said, is used for Amplicon sequencing analysis. So we might actually want to talk about what that means, right? So in microbial ecology or microbiome sciences, a lot what we do is based on sequencing data. So we basically run like DNA from an environment through a sequencer. And there's basically two approaches you can use. The one we actually look at today is called Amplicon sequencing. And maybe what's a bit different from like whole genome sequencing or any other sequencing is that in Amplicon sequencing, we actually only sequence a really small part of a particular gene. And usually that particular part of that particular gene is supposed to be really informative about what organism, for instance, that comes from. It's often called a marker gene because it's a marker for a particular condition or also for a particular taxonomy. For instance, you know, you could actually choose, if you would find like a particular part of a gene that varies a lot between different species of organisms, you could use that to distinguish different species, which is often something you might be interested in, right? You get the mixture of DNA from an environment. Now you want to know what organisms are in there. You actually need to sequence something that gives you that information. And if you want to save money, you probably want to sequence the smallest possible part of the genome that gives you that information, because the longer the part of the genome is that you want to sequence, the more you have to actually spend on sequencing. So the idea is to have that marker gene. And often the design we want in a marker gene is that it has some very conserved regions. So we can actually design a PCR primer that connects to it. And then it should be followed by a really variable region that we can actually sequence, and that will be, you know, very informative for us. So the basic step for Amplicon sequence is kind of like, you know, you do a DNA extraction from whatever environmental or host sample you have, you do a PCR amplification of the particular Amplicon only, and then you sequence it. The contrast to that is shotgun metagenomics. So Amplicon sequencing is not metagenomics because we are only actually looking not even at an entire gene. We're not even sequencing an entire gene. We're only sequencing one small part of one single gene. So, you know, it wouldn't be genomics because genomics looks at the full genetic material of an organism, not like the small part of one single gene. So the alternative would be shotgun metagenomics, where I actually don't really sequence only a small part of it, but I actually sequence the entire genome of all the organisms in the environment, basically using random primers. And that is usually done by DNA extraction. Then you do a fragmentation where you kind of break those genomes into really small parts. You'll ligate some particular adapters to it that will allow you to actually sequence all of that, and then you put it into the sequencer. And that will actually cover the entire genome with really small reads, which is pretty different from the marker gene, where often I will actually choose the variable region to be kind of in the length order of a single read. So like you have one single read kind of covering that particular region, or, you know, paired end reads covering it from both sides. So it's kind of obvious that for shotgun metagenomics, you will need much more reads to kind of cover the entire community, right? So if you have like 10 organisms in here and you do 30 reads, well, you kind of cover each of those variable regions like threefold if all of the organisms would be in the same abundance. For shotgun metagenomics, the full genome, bacterial genome is in the order of 1 to 2 million base pairs. With some variation to that. So like that's definitely like we need way more sequencing to actually get to cover the entire genome. So what is actually a good Amplicon that we could look at, or what is the gene you want to use for that? We will use the 16S gene. The 16S gene is not encoding for protein, and it codes for a ribosomal RNA, the 16S subunit of the ribosome. And it's basically the part of the ribosome that actually connects the ribosome to an mRNA to initiate translation. So it's kind of obvious that most organisms will have that gene because, you know, you kind of do need the ability of your ribosome to connect to mRNA. That's pretty ubiquitous to life. And why is that actually a perfect gene? Because it has exactly that design that we wanted. It is a pretty conserved protein for like most part, but it has those really, you know, hyper variable regions interspersed into it. And, you know, you could explain it with like for a bacterium, like depending on the environment, the way it actually the bacteria initiate translation, and also, you know, with what velocity, it's obviously like very organism specific. So you will have those variations there that kind of dictate, you know, how you initiate that. And then you have those interspersed super conserved blue regions here that are like perfect for primer design. And like the most common variable region in the 16S gene is the V3 and V4, especially for bacteria. Also because they're fairly short, the V3 is a bit shorter than the V4. If you have longer reads, you can actually cover both, but it's kind of really nice for the primer design because you can actually like, you know, target your primer to hit those conserved regions and then kind of sequence and cross the entire variable region till the end. And since you actually, you know, like I said, it's fairly easy to cover like a single variant or a single 16S gene, single Amplicon from the 16S gene quite well with only relatively few reads. What we usually do is in a single sequencing run, we can actually, you know, integrate a lot of different samples. So you don't have to, you know, run a sequencing run for a single sample, but you actually also adapt like a small sample barcode before the primer that kind of marks from which sample that particular read will be coming from. That way you can actually mix all the samples together because they're now all marked with that specific barcode here. And then we can pull that apart later on. And that's exactly the demultiplexing steps that I introduced in the beginning. So, okay, that's a lot of background right now, like maybe you're getting itchy to actually start analyzing something and actually get real data. So what we'll be looking at today is data from the Earth Microbiome project. And I should note that today and also tomorrow, the data that we analyzed is not something like that analysis is not already done in a paper. It's something that we will do only in the course. So whatever you discover is kind of new. So that will be exciting. So the Earth Microbiome project was a really large endeavor, and Sean Gibbons, that gave the introduction this morning, is a co-author on that paper, where basically the goal was to kind of look at the microbiome on a global scale. So they sampled the microbiomes from a lot of different regions on the planet. It's not sampled from everywhere. It's like representative regions, and actually like new iterations of the Earth Microbiome project have now sampled from way more regions than the initial paper, but still it was a pretty large endeavor where they sequenced like thousands of samples from different regions. So for instance, from the animal gut, but also from like the oceans, sediment, soil, all of that, and they analyzed those samples. And like, created some really cool insights, and also, yeah, had some conclusions how you should actually analyze that data. So that's kind of data we will look at. We will not look at all of the samples because that would be a bit out of the scope here, but we have like an example data set from, I set up, I would say, 5 fairly different environments. So we will actually analyze the microbiome from the honeybee gut. We will look at the microbiome in the cenotes. Cenotes are sinkholes mostly on the Yucatan Peninsula. Those are really deep sinkholes connected often by underground like cave systems. It's a really cool and interesting environment as well. We will have a few samples from the ocean. We will have a few samples from the human gut, maybe the one that is like, most common to all of us. And we also look at a few samples from the soil. So for each of those environments, we have 3 samples each. So we get 15 samples in total. So what's basically what we try to do today? Well, we actually run through a full analysis. So the steps we will run through is basically we start with the raw sequencing data. We will kind of quality check it and quality control it, maybe remove a few of the reads that have really low qualities. Then we will do a step which is called inferring sequence variants, and we will talk about that later. But the main idea is that a sequencer generates errors. So not every read that you actually get from a sequencer is the actual sequence that you had in the sample, but what we actually are interested in is the actual sequence of the 16S amplicon. So there's a step to actually recover those sequences. We will also assign taxonomy to it and actually see, which sequences pertain to which organism. We will talk a little bit on how we have to normalize the data to actually analyze it. And then we will do a lot of diversity analysis and also generate a few ecological insights. So let's maybe talk a little bit about, what does that sequencing data actually look like when you get it? So let's say you sampled something from, a super interesting environment. For instance, the honeybee gut. You had your honeybee gut microbiome samples, you pass them on, you extract the DNA, you pass them to the sequencing unit. What you will get is something like that. You will get a file that is called like that. It looks super weird, right? But let's maybe walk through the file names. That's what you usually get from Illumina sequencing. So it's a pretty common way of naming it. So the first part is usually a sample ID, and hopefully that's the sample ID that you pass on to the sequencing unit. So that's something that you provide. Here it's a bit of an odd sequencing sample because it was taken from the Sequence Read Archive, which is like a global repository for sequencing data. And that's like the ID of the particular sample. You get like a sample order. That's usually, it's kind of just like an index of the sample, 1 to the number of samples that you had. And it usually also corresponds to, what order they were actually injected onto the sequencer. You often get something like a lane here. Sometimes sequencers run like in parallel on several lanes to kind of, to have a bit higher throughput, and it will tell you from which lane it comes. And then you will also get the read direction. So you can have single end sequencing where you only sequence a single read from like one side, but a single fragment from one side. But you can have also paired end sequencing where, for instance, I cover like a variable region from both sides. So I start from the start and then also sequence it from the end. So I get those paired reads and kind of overlap a little bit. So that will tell you what direction the read is from. And then it usually has like a 001 at the end, which never changes. That has no meaning. And it's like some like compressed version of something called a FASTQ file. So a FASTQ file basically means faster plus quality scores, and faster is basically just a way to save sequences. And it usually could look, for instance, like this. That's actually like one part of the file, what it would look like. So you get like an @ followed by the name of the sample. Then you get some weird number, which often the number of the reads, because you get a lot of reads out of a sequencer. Modern sequencers can easily generate 1 billion reads per single sequencing run, and it tells you the length of the read. Maybe in this case that's the read that is 250 base pairs long, and then it actually just follows up with the exact read. So that's like the DNA sequence that the sequencer got out of there. Then it has a small plus, and then it has like a really weird string here of letters and numbers, and that actually encodes the quality of each of those sequence positions, because the sequencer itself knows it makes errors, so it will kind of try to log, like if a particular position in that read has a high or low quality. And basically it doesn't use numbers for that, because if you write like a quality score of 10, it would be 1, 0, so it would overlap with 2 of those nucleotides here, so you wouldn't be able to tell, is it like the quality of 1 for the first one and 0 for the second, or is it 10 for the first? Like it would all be shifted. So basically what they do is they encode every quality score, and quality scores usually go from 0 to 40, for instance, like with a single unique letter. So for instance, you could say that B is lower quality than C and D is higher quality than C. And then they put that underneath the read. So now it's kind of obvious, that this first nucleotide has a quality score of B, which might, substitute into a quality of 20, for instance. So that's kind of the data that you get. And it's not really, you won't be able to make sense out of that initially, right? So let's kind of get that into Qiime now and actually, generate our first artifact, and it kind of creates like a chicken egg problem. I already said that the only thing that Qiime knows are artifacts. Qiime can transform artifacts into other artifacts or artifacts into visualizations, but we don't have an artifact, we have sequencing data. So we have to kind of create an artifact from that sequencing data first, actually use it in Qiime, and that's what we will do now. So let's maybe go back to our notebook. Let's move this around here a bit in my own copy. Luckily, I still am connected to the notebook. That's great. So everything is fine. And if I look at the Qiime 2 installation, I actually see that it finished. So it took about 8 minutes in my case. That again is what we will do today. And like I said, the first thing we want to do is actually get our sequencing data in there. And the way we can do that is there's a few different ways to import data into Qiime. But we will start here with one which is called, sequences with qualities, and that uses a manifest format. I think that's like a really nice general format that works for most cases. And the idea is that, for your data input files, you actually write a manifest, which is like a tab separated file. And I can show you how that looks like. If you go in the data folder, it actually has the raw sequencing data. So that's kind of, those FASTQ, compressed FASTQ files that we just introduced. And if I look at that manifest file, it's actually fairly simple. So it just gives us an ID and then gives us an absolute file path, which, that just means the current directory / data and then that file path. And that can be quite nice for a project because you can kind of, keep track of which files you actually want to include in analysis. So you have an acronym list, and also, after you've done all your analysis and you might have to upload it to like a public repository, that manifest will be super helpful to know what to upload. So we have that list, we tell it where all the files are, and we say that we want to generate an artifact. And artifacts in Qiime are usually called like QZA. That's like the file ending that we want to generate, which is like Qiime zipped artifact, because those are actually just zip files that include the artifact and some annotations on how they were generated. So we can run this, which will start importing the data now. And while it does that, let's maybe look a little bit like how that looks like what is the format of that comment, what are we doing here? So anything that we do in Qiime will start with Qiime, and we have that exclamation mark here because the Colab notebook by default wants to run Python code. It doesn't really know that we want to run code from the command line. So what we have to do that is to start with an exclamation mark. That's a way to tell Colab, okay, that's a command line comment, it's not Python code. And then it always starts with Qiime. Then you basically have like a plugin or like a sub part of Qiime, which in our part, we use the tools sub part, and then you need to specify the action. In our part, the action is import. It will ask for a type here. Like I said, Qiime has a type system. So we would say that we have, we have sample data from samples, and those are sequences with quality. There's also paired data if you have like, and things like that. We will have to specify an input path, and we have to specify an output path, and all of that always starts with 2 dashes. So the 2 dashes are kind of, parameters or specifications that you add to that action, where you actually tell it, you know, what to use, what are my inputs, what are my outputs, what is the format. And then here we use the single end FASTQ manifest, and you know, you don't have to worry about the thread part here. That basically specifies in what formats the quality scores are. It's almost always that particular version here. And that's kind of like summarized here. So a Qiime is always like the Qiime, then the plugin action, and then you specify the arguments. And the arguments usually have a specific naming, so it makes it a bit easier. So if it's like dash dash I dash, it's like input files, dash dash O would be output files, dash dash P are parameters of the analysis. We will look at that a bit later. And dash dash M is metadata that I add in for an analysis. So we have generated the sequences here into, we do have an artifact, so you can actually look at your folder. If it doesn't update, you can also click that particular refresh button here, which will kind of update it. And we see that we have that sequences QZA file, which is like 80 megabytes large in our case. Okay, so now maybe also let's follow that up and actually generate our first visualization as well. So our first visualization, we will use the Dmax plugin. We actually will not demultiplex the samples because we already have one file for samples, but we use the summarize action from that particular plugin, which allows us to take like, you know, a sequencing artifacts and generate like a visualization of the sequencing qualities actually, because we have those quality scores. So one thing we might want to look at is like, you know, is that like good sequencing data? Did something go wrong during sequencing? That's probably the first thing you will do. So let's have a look here by generating that. And we'll take maybe a little bit, but not too long. And it will basically generate our first visualization. Visualizations usually are saved into files ending with QZV, Qiime zipped visualization. But now, well, it is something graphical, but how do we actually look at it, right? So the way you can look at it is by downloading it and then opening it with a viewer that Qiime 2 provides. That's probably the easiest for now. So you can click on that link and it will actually open that viewer here where you can actually drag and drop the visualization. It will tell you, you know, drag and drop it here. And let's maybe do that. So you can go on that file, you can go on that little 3 dots on the side of it, and it has that download option. So let's download it. Whoop, I see it here. And now you can basically go on here, click on it and choose it. Should be in downloads for me. You have that qualities visualization file. You can open it up and you see something like that. Most visualization in Qiime will basically be in a format of like a web page that kind of combines, you know, text and maybe also the figures for it. So we can actually look at, you know, our entire sequencing data here a little bit and get some summaries. So what we see there's only forward reads. We don't have paired end data. It's single end data. So it's only sequenced from one direction. We see the smallest number of reads we have for a single sample is 20,000, which is more than enough. Usually, you know, a rule of thumb for like Amplicon sequencing data is about 10,000 reads per sample. It also depends on how diverse the particular environment is that you sequence. If you have like 2,000 different species in an environment, you probably want a bit higher depths and more reads. If you have only 2 or 3 species, you can probably get by with a few less. But in mean, we actually have 70,000 reads per single sample. So that's pretty good. That will definitely cover most of the organisms in the sample, and it goes up to 200,000. In total, we have about 1 million reads in here. And then you will basically see that also for each of the samples. But maybe more interesting is to actually look at the interactive quality plot here, which will actually summarize for all of the samples, like what is the quality per single position. So you can actually see that, you know, qualities here range from 0 to around 40, like I said, and you will actually have a maximum of 40. That's actually like a logarithmic scale. So one thing that I remember is usually by is that like the 10 is like a good reference value, because like a quality score of 10 means about 10% of error. That means like for every 10 bases that you would sequence with a quality score of 10, it would make 1 error. But 20 actually is like an order of magnitude less. So like 10 is 10% of error, 20 is 1% of error, 30 0.1% of error, and 40 is 0.01% of error. So that's like very low error rate. And you actually see that, that's the entire sequencing read. So 0 is kind of where you start sequencing the single read. It goes up to a length of 150 base pairs. That's the length of our reads for those data set. And you see it kind of summarized in those mini box plots. You can also, if you wanted to, you can zoom in into specific parts and double click on the image to go back to the entire view. And you see that, for most parts, they're a little bit lower maybe in the beginning, but still pretty high. Along the read, they're fairly high, but then you see that dip towards the end of the read. That's pretty characteristic, especially with longer reads, like 250 base pairs, you will see that dip way more pronounced. And then they get into the danger area. So the 10% error rate, that's pretty high. 10% error rate is where you would say that, oh, maybe I don't want to deal with that in my analysis. So that's where you want to trim the read, maybe like cut it off a little bit earlier before that to actually, only keep the nucleotides that are fairly high quality. In our case, the quality is pretty good, but like we see, especially it's the last base here, the 150 base pairs, it actually looks, it's suboptimal. It's not high quality. There's definitely a few samples that go below that, the quality score of 10. So that's something we might want to trim. You can also trim a little bit earlier if you wanted to, if you kind of see, I feel like around 140, it's kind of, getting a bit lower, or maybe a bit earlier, you could do that as well. So that's kind of, just to check out the qualities. And they will actually need that quality score for the analysis that we follow up with, because now we kind of go into a single plugin, which is called Data2. In Qiime 2 that will do a lot of things, and it will also take a little while. So basically what we will do is we will, just summarize what it does. We will start running it, and then we will actually go into the weeds of what's actually happening here. So the Data2 plugin will do 3 things, 4 things actually, if you want to add that in as well. But so basically we'll start with filtering and trimming the reads. So basically it will go through the reads. You can specify if you want to cut off the end of a read. Like I said, if we want to, for instance, in my case, for instance, I might have decided that the last 10 nucleotides of the read is like really, I don't trust the qualities that much. So we'll actually cut the reads all at the nucleotide position 140 at the 3' end to get a bit shorter read, but with higher quality. It will also go through and remove reads that have like a very low average quality. So it will basically, if you have a read that, for some reason didn't really sequence that well, it will also remove those. It will actually find the localized set of unique sequences in the samples, like I said, and we see that also in the qualities. Here, the sequencer doesn't have perfect quality. It does make errors while it sequences. So if you would have a single 16S Amplicon in your sample, you might still observe thousands of different reads. Like they will all differ maybe in one nucleotide position on 2 of them, because that's the sequencer isn't 100% accurate. It does make errors. So we definitely want to kind of go back and find those original sequences, right? If I have those like thousands of reads, I might be interested in seeing, what is the actual 16S sequence like that actually generated those reads. We remove chimeras, and we will see in a minute what that's about. And then we will also count the abundance of each of those unique 16S amplicons. So for instance, if we have one particular amplicon that is of E. coli, for instance, we would like to count it because, there's some assumption that if you have more reads of that particular thing, then probably was more abundant in the original sample. So if we have more counts of a particular 16S amplicon, that organism was probably more abundant in the sample, roughly speaking. Okay, so that's like, again, it's the plugin is Data2. The action is the denoise signal. Denoise basically means, remove the errors, so to say. Our multiplex sequences is the sequence artifacts that we generated. I truncated here at like a length of 140. You feel free to change that if you want to try out maybe a bit longer reads. We will use 2 threads because in that particular environment, we have 2 CPUs available. And instead of really like specifying the name of the output artifacts, because that particular analysis will generate a few of them, we actually generate an entire folder called Data2, and we run it in verbose mode, so it actually tells us what it's doing in each step. So like while that will start now, it will also take about 10 to 15 minutes. And there's like a lot of output. You don't have to be scared of it. It's, just informative. There won't be anything bad happening. it will start by reading in the sequencing, and now, for instance, starts filtering. And while it's running that, let's talk a little bit more about what's happening here. So we have our analysis. We brought in, data to and like kind of started our analysis. So like I said, it will filter and trim the reads and all of those steps. And the pre-processing is the first thing that it does. And like I said, it will trim the low quality regions, but we actually have to specify and choose them. It will also remove reads with low average quality. It will remove reads with ambiguous bases. So sometimes the sequencer doesn't even know what nucleotide is actually in that position. It will just assign an N for like, it doesn't really, basically marks like an unidentified nucleotide. So those things usually mean that we didn't really, work out really well. It also removes phi X. So that's usually, since the 16S genes, like I said, is fairly conserved, and we're sequencing basically the same thing, like from a lot of different organisms. A lot of sequences have problems with there's like that little variability in the sequences that you sequence. So you add in actually a genome of a really small genome of a bacteriophage phi X that we know perfectly well. And we just add that in to create like a more heterogeneity in the, the sequencing reads, and that has to be removed afterwards. Like the sequencer needs it, but we have to filter that out. And then maybe the base step or the core of data 2 is to identify those alternative sequencing variants. So that was actually a major shift in the field. Before that, we mostly, what we did is we clustered reads together. So we would say that if a read is pretty similar to another one, we put it into a single cluster, and then we would kind of say that this cluster might be generated from the same original sequence, even though we didn't really know that sequence. And we would create those groups, and that was usually called OTUs. But yeah, a while ago, like there was that movement to what we call an alternative or Amplicon sequencing variants, where we basically have an algorithm that can actually infer the original sequences that generated all the reads. And the basic idea is like a kind of a two-step process. You kind of know there was an original sequence that generated all those reads with small errors. And that happens for all of the original sequences in the sample. So now if I get like any sequence, my question is like what actual sequence was it generated from? Where are the errors, right? And one thing that this algorithm does is it kind of, splits that problem up into two parts. So the first part is, if I would know perfectly well, like how like a quality score from the sequencer really translate into probability of making an error for any particular substitution. So there might be a misread from A to T. So there might have been like an A in the original sequence, but it was read as a T by the sequencer. Might also be a misread from A to G. And those don't happen necessarily with the same probability. It also really depends on the sequencing run and the sequence itself. If I would know that error profile really, really well, then identifying the original sequences wouldn't be too hard. So I could basically, for every sequence, look at how probable it is that that one sequence was generated from another, and that way I can identify that. But I don't really know that error profile, so that kind of doesn't work. The other way around, if I would actually know the original sequences in my sample, I could easily generate the error profile, right? If I would know the sequence of that particular sequence 2 here, for instance, I could just look at all the substitutions that were made in the reads and kind of, estimate the error profile. And then like the way they resolve that, which was called an expectation maximization algorithm, where they basically do those 2 steps again and again until it converges. So basically you could start with like a random assignment. You could say, oh, the most abundant unique sequences. Like if one sequence has like 1,000 reads that look exactly the same, maybe that was an original sequence of a 16S amplicon. I do that, I estimate the error profile. And then in the next step, I actually assign new original sequences based on that estimated error profile, which is probably wrong, right? Because, it's kind of like a, imperfect argument to make, like that those are the original sequences. But the thing is that in the next step, my estimate with those new original sequences and the new error profile, I get closer and closer to the real one. And like, the cool thing is, and it's almost like magic that this always works, we'll always end up with the kind of the correct error profile and the correct Amplicon sequencing variant. So we'll actually recover the original sequences that generated all those reads. So you might in a sample C, you have like, 20,000 different reads, but they will actually collapse to 50 different Amplicon sequencing variants that have like a lot of counts for each. You would see the first sequence variant has like appears 100 times and things like that. So that's basically the trick of data too. One thing you have to fix afterwards is you have to also, you know, account for PCR chimeras. Like I said, we're sequencing like kind of a single gene that's like very, well, a single amplicon that's very similar still across organisms, even though we have set variable regions in there. But one thing that often happens during the amplification with PCR is that basically you get some aborted extensions. But since like the first part of your initial, you know, we start with a conserved region, right, in every primer, and it still like goes into a little bit into the actual read. So you get those mispriming events where a new sequence is similar enough to still prime with that aborted extension, and it will just, you know, continue elongating it and filling it in. So you basically get a read that is like a mixture of two things in the original sample, which is like a chimera. We have to remove that, and there's an algorithm for that as well. So that's kind of another thing that data 2 will do. And we can kind of check back in and still learning the error rates right now. So let's maybe go ahead a little bit more. So we now have a table containing the counts for each ASV in each sample. So we now basically have recovered the original sequences or the amplicons that were present supposedly in the particular sample that we analyzed. We also have a list of the ASVs. So we actually have like the actual sequence of that original sequences, and we have like how often they appear in each sample. So now you can maybe like post in the chat, do you have any idea what you would do only with that data? So we still don't really know what organisms those things belong to, right? So we only have like a sequence, which is like the original amplicon sequence in the particular sample, and you might have a count. So in sample 1, it might appear 100 times. In sample 2, it might only appear 10 times. So you can like post in the chat and kind of see, what would you do with that? Like, what would you be interested in with, if that's the data that you have available? Maybe, you know, if the TAs see any, they can tell me what the chat is saying. The question is like, you know, the information that we have right now is we have the amplicon sequence variants for each sample, like we have the counts, and we also have their sequences, right? So what would you be interested in looking at only with those? Like if you know that a particular amplicon appears a lot in one sample, not in the other, like could you like think of an analysis you want to do with that? Okay. Yes. Variability between samples, diversity. That's a great, you know, those awesome points. So yeah, the very first thing is, even though we don't really know what organisms they belong to, yeah, a really basic question is how many of them are there actually in one sample? How many different amplicons, right? Because if our assumption is roughly that each amplicon comes from a different organism, it's not always true. Like sometimes a bacteria can have several 16S genes and they might be a bit different, but you know, roughly speaking, it's kind of true. So if I see 100 different amplicon sequences in a particular unique amplicon sequences in a single sample after we run data 2, but I only see 2 in another sample, that like, you know, goes into like a question of diversity, definitely. And also variability is a really good question. Like, you know, why is one ASV in one sample and not in the other? That means that it's fairly specific. So maybe it belongs to an organism that only thrives in a particular environment. Definitely. So that will run through the data 2 comment here. And it will finish pretty soon in my case. And probably for some of you might already have finished. In case, like, you know, it doesn't really finish for you, or you don't want to wait, you can also stop it and you could run the next cell, but, you know, better wait until it finishes to actually, you know, apply all the, if you've changed, for instance, the truncation of the length of the read, then it will be like you will actually get like different data from the treasure chest because that was generated with a blockade. But now, for instance, in my case, it finished denoising the samples. Now it will only go into removing the chimeras, and then we're basically done with data 2 because then it will only assemble the counts, which is fairly quick. But yeah, to maybe continue a little bit while that's still running, like exactly, I think a good question is about diversity. And actually, both of the concepts that we'll be looking at were already addressed here. So there's two different types of diversity metrics or ecological diversity metrics you might be interested in. The first one is alpha diversity, and that's the diversity within a sample. So it's, for instance, related to the question of how many different things are there in a different environment? How many different organisms are there? Or are they really equally abundant or not? The other question that was mentioned here, variability between samples, that's related a little bit to better diversity. And that's not, within one sample or with one environment, but it's between different environments. So ecological difference between samples. So that's something we might want to look at as well. So we want to compare if the composition of Amplicon sequencing variants in one sample is kind of similar to another sample, right? So those are definitely good questions. And in this part, it's also done. So it actually generated a few different things. We have we get a table, which is basically the counts. We get the representative sequences, which is the sequences of the particular Amplicon sequencing variant. And we also have some denoising stats, and that's all saved into a folder called data2 here. So if you open that up, you actually get that folder that has those three artifacts. They're still artifacts. There's nothing we can really look at. We could if we wanted to, but maybe we'll just go ahead with analysis. And one thing I want to kind of discuss here is, we looked at single-end sequencing data, so we didn't have to worry about that. But, one of the, for instance, most asked questions or general problems that people have on the Qiime 2 forum is kind of losing a lot of reads in that analysis. So they might run with paired end data through data2, and they suddenly see that, in the merging step where it actually overlaps the two reads to generate the full, like a longer Amplicon, it kind of nothing gets left. And that has usually something to do with, very aggressive trimming, which can sometimes really break up those things. So basically what happens is exactly, so that's, for instance, here you have like the V4 regions, which is the same region that we sequence in our example. And we actually do paired end sequencing, which kind of looks like that. So we have the read 1, which actually sequences from one end, and read 2, which sequences from the other end. And ideally they overlap with a certain amount. Usually that overlap is not really large. It's like somewhere between 10 and 20 base pairs for like most analysis. And now I might actually do the quality analysis, and I might think that I'm doing really good because I will actually be like super aggressive. I will cut off a lot of the end of each read to kind of really get really high quality reads, which, intuitively seems like it makes sense. But then you might actually get into a case where you cut off so much of the trimmed read that now the overlap is completely gone, right? Because that's basically both ends of the read overlap. So if you trim off the ends, you might actually lose that overlap. And one thing that data2 does is will actually always infer sequencing variants separately for forward and reverse reads. You should actually not pre-merge reads and then pass them to data2. And in Qiime 2, that's not even possible. So that's by design. But then after it actually infers the unique amplicons, it tries to overlap those amplicons again to generate the full sequence. So for instance, in this case, you get the full V4 region that was sequenced. And that if you trim too aggressively, then you might not have enough overlap. So there's definitely a trade-off, like, how aggressive you want to be when trimming off low quality ends from the reads. You should always kind of calculate what you can do. So a good example is if your amplified region, your amplicon is 450 base pairs long, and you have 2x250 base pair reads, which means, you can cover a region of a little less than 500 base pairs in total if you wanted to. So that's fine. If you overlap them, you kind of see that, you can overlap up to 25 base pairs of those and still kind of cover the entire region. So that's good. But then if you trim the last 30 bases of each read, you truncate the length of each read to 220 base pairs. So the length of covered sequence is, 2 times 220, which is 440, so that's shorter than the actual fragment that you want to amplify. So if you trim off too much, you lose that overlap, basically. So what you can do as well is, in case, you design very little overlap into your sequencing protocol, you can kind of adjust the p-min overlap. I think it's 12 nucleotides by default, but maybe you only generate an overlap of 8 nucleotides after you have trimmed your reads. You can actually specify that in that comment for data2 as well. Okay, so the denoising stats, like I said, are contained in an artifact. We can actually convert it to a visualization to actually look if, if we actually retained enough reads for further analysis. So let's do that really quickly. So we can run the next comment here, which is that Qiime metadata tabulate. So that's like an extra comment, which kind of what it does is it goes into almost any artifact that has like a table data, and it will generate a visualization that actually just shows us a table. So that's kind of like to get around that fact that like artifacts are usually not, you know, supposed to be used for visualization, but we can actually convert it and just look at the table. So let's look at that. So that will generate like a denoising stats visualization instead of from an artifact. So we can download this. And I will do the same thing again. And then we can go back to our Qiime 2 visualization and use it here. So I downloaded it in my notebook. It's kind of down here, the denoising, the QCV file. Don't download the QZA file because it won't show you the table. Then I open that up again. And you actually see for each of the samples that we had, we still don't know which environments they came from, but at least we kind of see, you know, what is the actual number of reads that were in the sample. For instance, 25,000 for the first one. After the qualitative filtering, we lost a certain part of it. So 20% of the reads. That's kind of okay-ish. It means that, you know, the quality in that sequencing wasn't great. That can happen depending on the environment, also where you isolate the DNA from. You sometimes get inhibitors or like other compounds and kind of interfere a little bit with sequencing. We see that other samples, for instance, get really high quality filtering. So basically only 1% or less of the reads are removed. Then we have the data 2 step, which might also remove a few reads, basically if it can't really assign them to any original Amplicon sequencing variants. So you kind of see that through each step of the analysis, you do lose a little bit of your input data. And then it will also tell you what is the non-chimeric output. So that's like the final amount in percentage. So we will see that, for instance, for the first sample, of all the reads in the sample, we retained information about like 78% of them. And around 80% is kind of expected in that analysis. Like I said, sequencing is not a perfect procedure. There's some odd things going on, so you might actually end up with less. Also, you know, you got 25,000 reads, but like a large fraction of those might actually come from chimeric reads, and we can't really analyze chimeric reads. Those are just, you know, an artifact of the sequencing. Okay, so in our case, that looks okay, so I would say. So around 80%, maybe 60, 50. It's not perfect, but if you look at the actual number of reads that we retain, like if we remember that rule of thumb that we want 10,000 reads per sample, we're actually above that for every single sample, so we're good. Nice. So now we can actually go and look a little bit at phylogeny and diversity. So as we motivated, we have that alpha and beta diversity, where alpha diversity is a question, you know, how diverse is a single sample? So how many different things do I have? So, and like we said, the most obvious one we could ask is what we often call richness. How many taxa do we observe, or how many different amplicons do we observe, which should be kind of related to the number of organisms in that sample. But that might actually ignore certain things. So, you know, I did those two examples here. Both of the first two cases here, like the left and the one in the middle, basically have 3 different organisms in them. But the second one, basically, even though it has 3 different organisms, it's dominated still by one organism. And that's something we have to take into account if we talk about, for instance, diversity, right? Because, you know, in one case, things are like very, very evenly distributed across 3 organisms. So that would probably be what we intuitively call, you know, the highest ecological diversity we can have. Whereas in the second case, we do have the same number of organisms, but 2 of the organisms are actually very low abundance in the sample. And that can be, you know, expressed by evenness, how evenly abundance is distributed across taxa. And maybe, you know, in the most ideal case, we actually want to mix that, right? We want to say, well, we consider something ecologically diverse if there's a lot of different organisms in there, but they are also very evenly distributed, you know, in abundances. And the most commonly used one is the Shannon index, which actually comes kind of motivated by information theory and physics, which kind of combines both of those measures into one. And we don't really have to go into the detail how you calculate it, but basically the Shannon index is kind of a quantification of how surprised you would be if you randomly take organisms from that environment. So basically in that first environment, I might, you know, get that rectangular organism here, and then I get a circular one, and then a triangular one, because they're all evenly probable. So I would be pretty surprised every time I draw an organism, even though in the second one, you know, even though I have 3 organisms, you know, I might draw a circle organism, then another circle organism. So I wouldn't say, well, I'm not particularly surprised by that since I already observed the circle organism before. And if I have like super non-diverse environment where I have like only a single organism, well, after I've drawn a few random organisms from that environment, I kind of would say that I'm not surprised anymore. That's like one interpretation of the Shannon index. But it's a score between 0 and, you know, up to fairly large values, which usually the higher the Shannon index is, the more diverse it is, and it takes into account the number of organisms, but also like how evenly they're distributed. So that's like something we might want to look at. And then for statistical tests, since alpha diversity is a single value for each sample, you can actually use any normal statistics. So for instance, you could use a t-test or Mann-Whitney U-test if you don't want to make that many distributional assumptions. To kind of compare that to, for instance, you can ask like, you know, are samples from one environment statistically, do they statistically significantly have a higher Shannon index than from another environment? And if they do, you would maybe argument that the samples from that environment are more diverse. Better diversity is a bit of a different question. Here it's about comparing different samples to each other and basically distributing them into like a space of closeness. So how different are two or more samples, donors, sites from one another? That's a question you would ask in better diversity. So for instance, here in the first sample, you could state that they're fairly similar because, you know, both of those samples not only contain the same exact sets of organisms, but also the abundances are not perfectly matched up, but they're somewhat like matching up. Whereas in the second comparison here, you would say that those samples are very different because not only do they have very different organisms in the sample, they're also like, obviously, like they don't even overlap at all. So they're kind of mutually exclusive. And there's basically two ways to do it. There's the unweighted one, which basically doesn't take abundances into account, how many tags are shared between samples. But also the weighted one would also take into account like whether they have the similar abundances. So for instance, in an unweighted score, I would basically say that the sample 1 and sample 2 on the left side would be perfectly identical because they contain the same organisms. But if I weighted by abundance, I would say they're not exactly the same because, for instance, you have the rectangle organism here has an abundance of 2, but only an abundance of 1 here. And if I compare, especially, you know, within an environment or across of different environments, I might also be interested in saying, you know, well, I might want to actually take into account that certain organisms can be really close to each other, right? So you could imagine a case where you do get a lot of different amplicons from a single environment, but if you look at those amplicons, they only differ by a single position in their actual sequence. So they're very similar. They're almost, you know, they look like they might be almost the same organism or organisms that are very closely related. You might want to account for that as well. And one way to do this is by actually constructing those phylogenetic trees. So those are constructed in an iterative manner where you kind of go from the bottom and you kind of see, you know, which two amplicon sequencing variants are the most similar ones. You group them together. You do that for all of them, then you group those groups together until you basically have everything grouped up, and it creates that kind of dendrogram or phylogenetic tree here where you kind of have the individual sequences at the nodes, and then they get connected depending on how close they are to each other. So if you have that tree, we can actually define a distance, and that's like the idea of the unifrac distance. And the idea is like to sample genetically similar taxa as well. And if you incorporate that in beta and alpha diversity, sometimes you might get slightly different results because you now actually account for like also phylogenetic distances between them. And the way that unifrac is calculated, the main idea is that you basically go through that tree and you have everything, like every amplicon sequence invariant across all your samples. Let's do it here only for the example of 2 different samples. And what you actually count is in that tree is you count like branches in that tree where you basically mix samples. So in this case, you see that those in the left example here, the kind of green or teal colored branches here kind of have symbols from the same environment, but then the rest is kind of like shared between, well, those individual branches here are only for a single sample, sorry. And then the black ones are always like shared. So there's different samples that are kind of united by that particular branch. Whereas in a single example which is kind of the case if 2 environments would have completely different amplicon sequencing variants, you would see that case where basically all of the branches are somewhat specific. So like every branch is kind of specific for a single sample. And then that would be like where we kind of say, okay, that seems like, you know, a strong phylogenetic distance between those samples because they all are completely different regions of that tree. And then the unifrac is just, you know, counting the branches that are unshared versus the total number of branches. So if the branches are always like unshared, like meaning they're from they kind of only unite like samples or amplicon sequences variants from the same sample, then I would kind of say that's like a high distance between the samples. And if it kind of looks like the first case where everything mixes up perfectly, that would be it. Okay, and then like, how would you build that phylogenetic tree? I mean, we will do it like in an example in a second, but basically we would, like I said, we would actually just look how similar different ASVs are to another, and then we join them into that tree by just usually multiple sequence alignment. So we kind of, you know, align them to each other and then unites this, the particular amplicon sequence invariants that are similar into that tree. So they're arranged by sequence similarity. Okay. Maybe at this point we can kind of jump back into the notebook and actually run the analysis. So why did I spend so much time talking about, you know, everything like alpha and beta diversity together? Because you can actually do that analysis almost in a single step in Qiime 2. The first thing we actually need to do is to generate that phylogenetic tree from our raw sequences. So that's kind of done in one single step, where we basically now use the phylogeny plugin from Qiime, and it has that action that is aligned to tree, and it uses, you know, some kind of a fast tree in the particular liner for that. We only will use the representative sequences because that's basically the individual amplicon sequence variants that we obtained, and we'll output that again to unfold a tree which will contain our particular tree. And I see that some people are asking about rarefication or rarefaction. We will talk about that a little bit later when we actually compare abundances between different things. But yeah, that's definitely something we have to account for. So we can first like generate that tree. When we have that tree, we can actually run the diversity analysis, but we might also want to take a quick peek of what that tree actually looks like. And that, you know, just to see, you know, do we have like very similar amplicon sequencing variants in general, or are they fairly different? And for that, we can actually use like a different plugin that got installed into that environment, which is called Mpress, which is basically just a Qiime 2 plugin to visualize phylogenetic trees. And that's what will be used to actually visualize the trees. And while that is running, maybe are there other questions that popped up in the chat repeatedly that we should maybe address?
There are several.
Okay, yeah. Rarefaction, we will talk a little bit about that in a few minutes or maybe after the break, since we're getting really close. That's definitely something we want to look at. And then, yes, if you want to know how many reads your samples have, that was actually one of the first steps, and that's why we ran that. You know, we generated that artifact from the raw sequencing data, and then we had that quality visualization file. And if you remember that, I can go back to that here while we're still analyzing and building the tree. If you look at the qualities here, it actually had that summary, how long the reads are, and you have that under the interactive quality plot. So if you check the quality plot, and then you go down here, you actually get, you know, this demultiplied sequence length summary. You will definitely always see like some reads are shorter than others, but you will see that, you know, at least the median or more than half of the reads will have a pretty specific length. In this case, it's 151 nucleotides, which is actually the target sequencing length by the sequencer. Well, 150 base pairs is what you kind of aim for, and you usually get like an additional base or something. Whereas some of the reads might be a little bit shorter, and you actually get the distribution of the lengths as well here a little bit by, you know, it's telling you that about 10% of the reads are like shorter than 130 base pairs. So you can use actually the same tool to kind of, look at how long your reads run. Usually, your sequencing unit should know, because that's part of the protocol that they use. So they know by default, like what's the target length the sequencer wants to go for. But yeah, definitely, if you get data from a collaborator, they might not be super sure. And it's always good to check. They might tell you it's 250 base pairs and you look at it, it's only 100, right? So that happens. Stuff gets mixed up, and that would be like a good analysis here. Well, that's a bit unrelated. They are Reveal.js slides, but they're basically a web page running in JavaScript. You can look at the GitHub repo. It has the base code for that, the source code as well. Okay, so we have that. So maybe before we go into the break, let's look actually at the tree itself, right? It might be interesting to see what, how those Amplicon sequencing variants compare to each other. So we run also the next cell, which is the Qiime Empress tree plot comment, and that will create a visualization. It doesn't have that much information in there right now because we only have our sequences, and we actually will have an exercise to generate a much more complex and like better looking tree, but start with that. So that will be saved into the tree folder. So let's open that up. So again, I go into that overview of files. I go into the materials section, look for the tree folder, try to find the visualization. That's the thing ending in QZV. And then I download it. And again, I put it into the Qiime visualizer for results. And those things can be shared, right, with collaborators. Those visualizations, you can tell them to open them up. So it's pretty convenient. Also, maybe I should note, like sometimes you might be worried that, if I upload my visualization here, like, is that lying on some server somewhere, which is not my own? Well, no, it doesn't. It actually is, everything is run in your own browser. No data actually gets uploaded anywhere. Your browser is kind of used like a magic machine that will actually, extract that zip file and visualize the web page. It actually runs all locally on your machine. Okay, so that's what the tree looks like. Looks funky for sure. Let's maybe zoom in a little bit. Oops, that was a bit too quick here. Sorry about that. And you kind of see it has those branches, like I said, it's kind of like the tree looks like. You can actually change the layout. So for instance, that's called an unrooted tree. So it doesn't really have that center node because like sometimes it's hard to identify it. You can force a circular layout, for instance, where it kind of, that's probably something you've seen a bit more regularly, where it kind of tries to find one particular branch node in the middle and it arranges everything circularly along it. And then if you look at it, you see those subregions here. Any endpoint of those is actually an ASV sequence. You can actually click on it and it will be very cryptic here. So you actually don't even get a sequence here. You actually get that weird ID. And that's because, Amplicon sequences can be fairly long. Actually, Qiime automatically generates unique IDs. So each of them is like one single Amplicon, and you could look into the representative sequences to map it back, but it won't give you that much information. What it will give you is to look at that tree, and you definitely see there's different clades, right? So you have those regions of the tree here that look fairly separated from the rest. And there's a lot of options here, and I will probably have you work more through that in your exercise. For instance, if you see that those branch lengths are somehow scaled with the sequence similarity. If they get super long, like in those cases, those means that those are sequences that are fairly different. So they're kind of different from all other ASVs. We don't really know what that means. We just know it's, it definitely has a very different sequence. If you kind of want to just see the general structure and kind of not be buzzed by those super long outliers here, you can also kind of say that you want to either ignore the branch lengths, which will make every single split the same length, or maybe more common is to make it ultrametric, which means that everything ends at the same length. And you get those plots, which you might have seen in a publication already, which kind of tell you how your ASV is distributed. And we definitely see there's some very large groups that are formed here. They were very early. We have like divergence into specific subgroups of the ASV. And there's a lot of more options you can actually do here. You can get statistics about the tree. You could export it as an SVG or PNG if you want to include it somewhere, but there's actually way more things we can do with it, and that will be done in the exercises. For instance, annotating the samples or the environments would be interesting here, and we will do that later on. Okay. So now, with 1 minute left before the break, oh no, actually it's a little bit more since we start a little bit earlier. Let's maybe start and just run the analysis, and then we can kind of look through it a little bit in the second part of the course and actually, but like having it run now, like we can actually wait during the break for it. So that might be pretty convenient. So we have the alpha and beta diversity. That's what we want to analyze. And like I said, there's like there's that really cool comment in the Qiime diversity plugin, which is called Core Metrics Phylogenetic, who does basically everything for you. So basically it will take your table of abundances that we got from data 2, which is basically the counts of each unique amplicon sequence variants in each sample. It will actually also take our tree into account, since it will calculate those unifrac distances. And it will do something, it asks us for something like sampling depths, and that's a bit weird. And that is related to the questions that people were asking in the chat. So the one problem that we have is, if we look, for instance, at alpha diversity, let's assume we have one sample, and for some reason, something didn't go quite well in sequencing, and we got maybe, you know, only 100 reads for it. Like if I have a really low abundance organism, it's very unlikely it will actually be covered by those reads, right? We have very few things that we actually sequence in that sample, so you might actually miss some organisms. It's the other extreme as well. We might have a sample which has like 1 million reads, so even extremely low abundance organisms will still get sequenced. So there's like a bias there, like really deeply sequenced samples might actually generate higher alpha diversity measures just because, you know, we sequence them with more reads. And you can actually really control for that really well. Like even if you're kind of really perfect in pipetting, and honestly, nobody is, like when you load it onto the sequencer, like you will always get variations across samples because it's basically kind of random. It's really such a nanoscale thing that the sequencer does that it often happens that reads from a single sample, like when it does the cluster amplification on the sequencer, get like pulled into larger clusters or something, which gives you a few more reads for a single sample. It doesn't really have any biological signal. It's like if you get more reads from a particular environment, it doesn't really mean there's more DNA necessarily in there. Like if that can still happen even if it isn't. So it's a bit unclear like what that means. So usually you want to account for that. There's like several approaches and all are kind of not perfect. The one that Qiime 2 uses here is called subsampling. So basically what they do is, to get kind of that effect removed, is they actually randomly pick 10,000 reads, or you can actually specify how much you want to sample from each of the samples. 10,000 is okay. Like we said, like it's like the minimum amount of reads we wanted for sample. There's trade-offs to that. Like the good thing is we actually account for, you know, that effect of missing certain organisms, because that subsampling in a really deeply sampled sample might actually remove some organisms just because of that sampling. So we kind of simulate the effect of missing organisms because of low read depths. The disadvantage is, if I have a sample with 1 million reads and I only do an analysis based on 10,000 random reads from that sample, obviously that might not be representative, right, for the entire sample. And even if I did it a few times, it might still be a bit odd. It's the same. So like there's definitely a loss of data involved here. So it's not, you know, it's not the only way to normalize that. We will actually look at different normalization technique at the very end, which is kind of, we try to, you know, transform the data in a way that maybe, you know, sequencing depths or like the sample size or the number of reads per sample is not really such a big influence on the value. But here, like by default, it's using that subsampling. We also put in a metadata file, and that's the first time we see that. Metadata file actually assigns metadata to your sample, so basically tells you what each sample is. And we can look at that as well. It's in the data folder as well. It's called metadata. It's again tab separated files. If you double click, you can look at it. And this one actually in our case matches the particular weird IDs that are not really informative to the particular animal or environments it came from. So for instance, you can see that the sample, the first sample here that ends in 680 is actually coming from the honeybee gut microbiome. And it will also has some more information, which is mostly sought for exercises as a project challenge. For instance, you can see which animal, if it's animal associated, like is it in an animal host? So for instance, for the honeybee and the human microbiome, that would be yes, whereas it's obviously no for the ocean, because that's not like an animal, and whether they come from a saline or non-saline environment. So we will put that in here as well to actually get like some nicer visualizations. And with that, let's finally actually start it. And that will run for a little bit. It will actually do a lot of things, and we will look a little bit at that after the break, I think. So let's maybe start with a 30-minute break here. And while we're on that, I'll also try to go into the Slack chat and see if there's some questions that still need to be answered, even though our TAs did an awesome job with that, as I've seen. And yeah, thanks so much. So we will be back in 30 minutes, and then we will actually look at our diversity data, actually try to make sense, get some ecological insights, and yeah, visualize a little bit of what is going on. So during the break, you will also hear a little bit from our sponsors. Like Sean said, this actually made that event possible. So we'll have a few short presentations and videos from them for you. And yeah, we'll thank our sponsors a lot for making all of that possible and actually having those presentations. Okay, see you back in 30 minutes.
In March of 2020, there was this ominous feeling around the world. Everybody was worried about how this was going to turn out. We had no idea how long this was going to last. There was a lot of discussions among myself and other leaders and scientists at Illumina, like, what can we do to help the world get through this pandemic?
Early on, during the first patients that were identified with atypical pneumonia in Wuhan in China, the China CDC connected with Illumina.
Now we have public health institutions coming to us and saying, hey, Illumina, we need your help.
Once we came up with the sort of strategy of how we wanted to do the COVID-seq test, everybody that we were talking with just jumped up, started getting some of these sub-teams to come back. You got to remember at that time, we were home and there was no clear plan for how you bring people back to the office.
So my son is immunocompromised, and so I was very worried about bringing this home too. It was emotionally very challenging, both on a work side and on a personal side.
There's not many times in your career when you can see something come together like this, and the teams that are formed, and you create a bond when you're responding to something that everybody has a passion and believes in.
In that time, that early parts, it was uncertainty, and it did feel good to be at a company that was going to play a fundamental role in fighting the pandemic.
The CDC brought up the idea, could we help them to respond quickly until they could get others up and running? And then here we are a few months later, and everybody wants to understand surveillance. So we had the foresight at the time to create an assay that we knew they would need before they even needed it.
Primarily, our intention was to participate in the testing, but as the pandemic has gone on, the value of sequencing and understanding the new variants has really become so important.
It was definitely very collaborative, very open, and it was really exciting to see people who were willing to put in the extra time knowing that it was something so critical to the CDC, critical to the United States, critical to the world in terms of tracking variants.
This pandemic has raised awareness about sequencing, about Illumina, and about the role it can play in health and healthcare.
All of you, from manufacturing to support to legal compliance, product development, marketing.
Thank you to everyone who worked on this.
Thank you for stepping up. Thank you for jumping into that crazy project.
Thank you for making sure that Illumina did everything we could to support laboratories and clinicians in fighting the pandemic.
We did this. We did it together.
It's a pleasure to speak today at the ISB Symposium, a place I love and that I've been associated with in various capacities for over 15 years. I'm going to speak today from a new position as CEO of Longevity, which is a division of Thorne Health Tech, and we are sponsoring the conference. So Thorne Health Tech is a company dedicated to personalized scientific wellness, which has been definitely a huge interest of mine beginning in my ISB days and working a lot with Lee Hood on a number of topics related to this. And the mission of this company is to bring the scientific rigor of biotech and pharma to the prevention space and extend the length of time that people can have healthier lives. We have two integrated brands that fit under Thorne Health Tech. One is Thorne, which is a natural product, high-end manufacturer and provider, and Longevity, which is an AI deep science division where we focus on delivering health intelligence behind scientific wellness. So here, what we're most interested in is microbiome for obvious reasons. We have a number of pieces that we've pulled together in this effort. The first is that we do metagenomic sequencing. That is through a test focused primarily on the gut called the Gut Health Test, which is available at thorne.com. We do machine learning based microbiome data analysis. We also work with groups, including via Longevity on clinical trials, design, diving in on details related to that. And then we also have a proprietary nutrition database that we link to via AI to link the testing to personalized recommendations that are also delivered via the program that we run via thorne.com. So we also do trials on the efficacy of the solutions. This is one example. I'll be very brief here. It's published in Precision Clinical Medicine. A lot of this is based around a lot of the science from one of our co-founders, Chris Mason, as well as some of the background that I have in microbiome research from ISB. Here we do clinically backed gut health protocol. In this case, was done for a 30-day trial, which showed that personalized interventions were able to reduce symptom severity scores for individuals with IBS, irritable bowel syndrome. And this was a highly personalized approach in the sense that there were 75 people in the trial and 68 unique combinations of recommendations that were used to achieve those results. One of the big things I wanted to talk about in the few minutes I've got today is around a new sample collection technology that we've developed, which we call the microbiome wipe. This has been, I think, a very gratifying thing to get to the market. But basically what it does is it replaces the way that we all typically do these microbiome collections, which is putting down that paper, pooping on the paper, having to scoop it, put it in a vial. A lot of participants, a lot of people don't love that step the way that us as microbiome researchers perhaps do. But what this does is it allows you to use actually just a wipe as normal. So basically no behavior change. And I'm going to show a little bit of the data behind that and take you through that experience, because we think this is going to be a much better experience for people going through and doing microbiome collection. And that's going to, we hope, increase adoption across the community of more microbiomes, which is good for all of us. So here's the bit of data. And if people want to dive into this later one-on-one, I'm happy to go through it more. But what this shows is that when we did the sequencing with the wipe, we actually got better correlation with a positive control, which is basically a shipped in dried ice gold standard positive control. And what we see is that if we compare this to the current technology, we get, in fact, better sequencing quality. If we look at the predicted measures like calculated scores or prediction of different metabolites and so forth, we find again that what we see on the wipe is actually a bit better than what we see on current technology. So we think we're getting both better quality in terms of being able to get sequencing and predictions, as well as also having a better customer experience. So participants, as they go through research studies that you may be conducting, there's a very simplified protocol for this then, right? You get some simple instructions. Basically, you wipe as normal, put it in a container, you shake till it's dissolved, takes about 10 seconds for that to happen. You release saline, which solution basically that preserves the DNA, throw it in a container, and you send it back. So that's all I have time to talk about in these 5 minutes. But the test is available at thorne.com for anyone who is interested. If you have questions, want to dive in on the science behind this, or are interested in partnering even on research studies, we do that both from the technology as well as computation side for those who need that. Feel free to reach out to me at nprice@longevity.com.